Okay, so let's do a little story. We're going to start off with two cells in G0 phase. So replication is not in the sites. Here's one cell. I'll draw the cell membranes as green, and then I'll draw their genetic material as blue. Like that. And so what happens is one of these cells undergoes necrosis. So it could be, mechanical damage, chemical damage, causing the cell to die. The cell dies through necrosis. So I'll label that necrosis. And from that dead cell is released some growth factors. I'll just depict that as pink. It'll just be a little pink dot here. And this surviving cell, well, it happens to have a receptor, or has receptor sites on its surface for growth factors. So that growth factor, or those growth factors, will bind to that receptor site on that cell. So let's label this growth factor. This cell dies, releases growth factors, binds to the receptor site on the neighboring cell. Okay. So this G0 cell, put G subscript 0 cell is recruited to G1 and then eventually transitions to the S phase. And we know what happens in S phase, that is DNA replication. DNA replication. Okay. So let's draw that cell again. This cell here. Cell membrane is green, genetic material, I think I used blue. And let's zoom in real quick into this nucleus, and we're going to be focusing on a single chromosome rather than drawing for every single chromosome in this cell. So that single chromosome we're looking at is going to be in chromatin form. We know that during interphase of the cell cycle, all of our genetic material is going to be in chromatin form. And so this squiggly spaghetti substance is pretty much equivalent to what we would see as a condensed chromosome that's one-lobed, a single-lobed chromosome. All right. So now let's draw what's happening to the single chromosome during S phase. And I'll just write it up here, S phase, S phase. Alright, so let's draw some DNA. here. So I'm just drawing the DNA in its unzipped form. So normally we've got two strands that are connected by hydrogen bonds between the nucleotides. All right, that should work. So something has unzipped this DNA. Something is unzipping this DNA. That happens to be an enzyme called helicase. I'm just going to draw that as black here, shade it in a little bit, label it helicase. And I'm also going to label a leading strand versus a lagging strand. I'm going to mark this as 3'. This is going to be the 3' end. And if we follow this up here, over here is going to be the 5' end. And then because DNA is anti-parallel, this end over here is going to be the 5' end. And tracing that up, that's the 3' end over there. So what happens is, as helicase is unwinding your DNA, and actually let's write that here on the side as a bullet for what's happening. So first, helicase unzips DNA. On this strand here, we'll say this is the leading strand. You've got this enzyme, DNA polymerase, DNA polymerase, let's label that DNA polymerase. What that's going to be doing is writing DNA in the 5' to 3' direction. So what it's doing here is it's creating new nucleotides, or not new nucleotides, but it's creating a new strand of DNA. And let's draw the backbone. And it's going to be writing it. It's going to write it, because it's anti-parallel here, in a 5' to 3' direction. 5' to 3' direction. So on leading strand, which I should probably label, let's see, I'll just write it right here, leading strand. So on the leading strand, DNA polymerase, DNA polymerase writes new DNA, that's pink, I'll underline that too, in a 5' to 3' direction. Okay. So now let's talk about this lagging strand. So before DNA polymerase can begin writing its new DNA, it actually needs to bind to an RNA primer. So we've got this other enzyme called primase. So we'll just draw it here. We'll make it triangular too. Okay, I'll label this as primase. And what that's doing is it creates an RNA primer. Creates a little RNA primer right here. And then from that primer, your DNA polymerase can start to build from it. So I'm going to draw that here. I'll just draw a DNA polymerase down here. And actually, before I draw the details here, let's talk about primase. So on the lagging strand, which this is, let's label that. Lagging strand. So on the lagging strand, primase attaches an RNA primer. And DNA polymerase from that primer will write new DNA. What color was that? Pink. In 5' to 3' direction. In the form of an Okazaki fragment. So let's draw the DNA really quick. So from here we've got, let's draw the anti-parallel part. And the backbone here, here's the DNA backbone. So I don't know if I mentioned this, but your DNA polymerase, what it's going to do is it's actually going to remove that RNA fragment and it's going to replace it with DNA. However, actually we'll get to that part in a second. So it writes it in a 5' to 3' direction, and I'll just label that here. So it's going to be 5' here, and it's moving in the 3' direction. And this is going to be creating what we would call an Okazaki fragment in the form of Okazaki fragment. Fragment. Okazaki fragment. Okazaki fragment. So here's an Okazaki fragment. Again, your DNA polymerase is going to remove that RNA primer. It's going to replace it with DNA, but you're just going to have the stretch of DNA from here to here. Now, as helicase continues to unzip this DNA, on the leading strand, that RNA polymerase is just going to, DNA polymerase is going to follow it until it's done. So you're going to have one large stretch of DNA. However, on this lagging strand, you have to make your DNA, your counter DNA, in a series of Okazaki fragments. So as this unzips it, you're going to have another primase create a primer over here, and then another DNA polymerase come to meet this DNA here. However, when it creates that DNA to meet this DNA, it doesn't fuse the backbone together. So there's going to be a little gap. So what happens is later on, you get this enzyme, and I'm not going to draw it here, but we get ligase. So another important piece here. So ligase eventually fuses Okazaki fragments together. Okay. So we had a cell in G0, cell next to it died, released growth factor, bound to that cell was recruited into G1, then transitioned to S phase. This happened to, one of the chromosomes. We can assume that this is happening to all of the other chromosomes. It's duplicating all of its DNA. So now let's draw the end product. So you got that cell, which we'll just say is in G2. We'll say G2. G2 phase cell. So it's moved through S phase, now it's in G2. It's got a nucleus still, but all the DNA is going to be in chromatin form. And again, we're just going to zoom in on that single chromosome. And we know that during all of interphase, your genetic material is going to be in chromatin form. It's just going to be twice as much at this point, that chromosome. Now this, if we were to translate this into a condensed chromosome, which it's not currently in, but if we were to translate it into it, it would look something like this. So here's the equivalent of a condensed chromosome, and now instead of it being a single-lobed chromosome, it is now a bilobed chromosome because it's undergone S phase. So this cell is going to move from G2 to mitotic phase, and then eventually cytokinesis, and then you have two, hopefully, identical daughter cells. One of these to replace the dead cell, wherever it may be.